Okay, so our first group are lipids. Okay, lipids, I want you to think of like fats and oils and things like that. Lipids are a family of compounds that have what are called fatty acids. I'm going to show you what those look like, and we'll get more into that here in a little bit. They are mostly, notice I say mostly, insoluble. So if you think of fats and oils and butter and things like that, they don't dissolve well in water. They're very nonpolar compounds for the most part, and that applies to most lipids. Okay, so here's some examples of different types of lipids. There's our waxes, fats and oils, phospholipids, if you think like phospholipid bilayer, if you're a biology person, sphingolipids, glycolipids, and steroids, which you are probably familiar with that word. So what they really are, like I said, they're compounds that have fatty acids, at least for the most part. Waxes are like long chain fatty acids, and long chain alcohols combined together, you get some ester bonds in there, right? Ester bond is C double bond O, and then another thing. Fats and oils, so notice these ones are starred. We're specifically going to talk about these three. Fats and oils are called triesters or triglycerides. They are when you have a glycerol, notice the OL on here, that means it's got an alcohol on it, OH, and three fatty acids bonded together. So this is a triester bond or a triglycerol, and I'll show you what those look like here in a little bit, but our organic tips are going to play in here. Phospholipids, as you might suggest, phospho means that you have a phosphate group on there. So instead of having three fatty acids on a glycerol, you have a glycerol with two fatty acids and a phosphate group with an amino alcohol. Sphingolipids are fatty acids with sphingosine and a phosphate group with an amino alcohol. So we're like substituting some of those fatty acids. A glycolipid is a glycerol still with a fatty acid and a sphingosine and a monosaccharide, which is a sugar. And steroids. You'll notice steroids don't have specific fatty acids, but they are a fused ring structure. Still very insoluble, and that's kind of why they fall under this category. So these are the three that we're going to talk about and identify. I'm going to show you what their structures look like. All right, so fatty acids are first. So that's the I don't want to say building block because usually you have more than one. You don't always have more than one in lipids, but a fatty acid is just a long chain carboxylic acid. So this is my example here. This is stearic acid. This is animal fat molecules. And notice it's a long chain, usually between 12 and 18. And then you have a carboxylic acid on the end. Okay, that is a fatty acid. So this is considered a saturated fatty acid because all of our carbon bonds are single carbon bonds. Okay, so when you hear those terms, saturated fatty acids and unsaturated fatty acids, it just means they're all single bonds. Or if they are unsaturated, they might have some double bonds in there. Okay, so there's mono unsaturated, meaning one double bond, or polyunsaturated, which means more than one double bond. So if you have a double bond in that chain, it might look like this. This is oleic acid, which is in like olives and corn. This is a mono unsaturated fatty acid. Or a polyunsaturated fatty acid might have two double bonds, okay, or more. And this is linoleic acid, which is the fatty acid in soybeans and sunflowers. So these are all natural. Unsaturated versus saturated just means it may or may not have a double bond. Okay. One thing that I want you to notice about these molecules is if they are unsaturated or they do have a double bond, they're usually cis, meaning that our groups go on the same side. So it bends in instead of like bending out. Okay, so if I get two, notice these are both cis, so this molecule is kind of like clumping up on itself. Okay, so those are fatty acids. Now I'm going to use those fatty acids to make these other molecules that are considered lipids. Okay, properties wise, I want to talk about the difference between saturated and unsaturated because they affect the properties of these bigger molecules. So this whole like zigzag pattern, if I have unsaturated, notice this is very linear. And when you have unsaturated fatty acids, they're really easy to like stack on top of each other and get them to attract each other with their London dispersion forces. So since they're like straight chains, they pack really well and they have stronger intermolecular forces. So they have stronger London dispersion forces and they end up being solid at room temperature. And so this one has a melting point of about 70 degrees. Now, if they have double bonds in there, though, you get some bends and things, and they're not as easily to pack together because you get different shapes and things. And so bent ones have lower melting points. They're usually like liquids at room temperature. They have weak intermolecular forces, and that's just because they don't stack very well. So unsaturated fatty acids don't fit together as well. They have weaker forces, and saturated fatty acids have stronger intermolecular forces. Okay, so how do we use these fatty acids to make other structures? So fats and oils, for example, are the ones that I'm going to talk about first. These are your most common types of lipids. Okay, usually when you think the word lipid, you think fat. And that's because they go hand in hand. So what a fat molecule is a glycerol. As you might be, what is a glycerol? A glycerol has 3 alcohols on it. That OL, right, means alcohol. This is a glycerol molecule. Okay, it is just 3 carbons. Each one has an OH on it. That's a glycerol. And it is bonded to 3 fatty acids. They do not all have to be the same fatty acid. They can be 3 different fatty acids. But each of these alcohols is going to bond with a fatty acid. And the bond is really just an ester linkage or an ester bond. Remember, an ester is C double bond O. So I have the C double bond O, and right here is going to bond with over here. Okay, so they're going to bond together, and then I'm going to get this nice big molecule, which is called a triglyceride, or sometimes a triacylglyceride, but a triglyceride. And this is my glycerol over here. It has made now 3 ester linkages or ester bonds, and I have my 3 fatty acids attached to the one glycerol. That is going to be like a fat molecule. And if you look up here in the right, this is a similar one, just a more specific example. I have a glycerol molecule. It's going to do 3 ester bonds. Right here, you take out a water, right, at H and an OH, and then I end up with an ester bond, C-double-N-O-O, bonded to the other carbon group. And like I said, they can be 3 different fatty acids. So I could have a bond here to caproic acid, to palmolic acid, to butyric acid, and this is still a triglyceride. It's just like the common name for these types of molecules. On to a couple other types of lipids. So I've got phospholipids and glycolipids. Now these 2 groups go together because instead of having 3 fatty acids, they only have 2 fatty acids attached to them via an ester linkage, and then they have something else on the 3rd alcohol group. So it's like a triglyceride, but one of the fatty acids is replaced in a phospholipid with a phosphate and a choline. Okay, so this is a polar head. This is a phosphate. Phosphate is a bunch of oxygens and a phosphorus. It's a very electronegative structure. And this is a choline. Okay, so this is what we call a polar head. Instead of it having 3 very nonpolar fatty acids, it has 2 nonpolar fatty acids. Okay, so this side of the molecule is very nonpolar. And then the 3rd thing that's attached is actually a very polar molecule. So this molecule is really big and has one side that has these 2 fatty acids that are nonpolar, and one side that has this group or this head that is polar. So a phospholipid looks like. A glycolipid is very similar in the fact that it has 2 nonpolar fatty acids, and then the 3rd group, instead of being a phosphate and a choline, it's a phosphate and a sugar molecule. Also very polar, a bunch of oxygens on there. So this is going to be its polar head. Okay, so when we actually draw out these structures just generically, we draw them like this. We have a polar head and it has 2 nonpolar tails. And on a molecular and atomic level, it looks more like this. Okay, so we have a polar side and a nonpolar side. And these are the types of molecules then that are used in the phospholipid bilayer of a cell. So its properties are very important for cells to be stable. Okay, this is a phospholipid bilayer, right? That's the layer that goes around all of the cells that are in animals. And these are our phospholipids. So they have a polar head and they have a non-polar tail. So the polar heads go on the outside and then the tails go on the inside. Reason being, our body is made up of mostly water, right? So the polar things are going to attract to the water. So the polar heads are on the outside attracting to water. The inside, however, is where all the non-polar stuff goes because it doesn't attract to water, but they do attract to themselves. So the non-polar tails go in here. Okay, so this is the phospholipid bilayer, okay, or lipid bilayer. It's the cell membrane, polar faces out, non-polar faces in. You'll notice there's also some glycolipids or glycoproteins here because they have that non-polar and polar side as well. Okay, now steroids. This is the 3rd group that I want you to know about. And they are any compound in the body, really, that has this structure in it. Now, it could have variations on this structure, but it's 3 6-carbon rings and a 5-carbon ring all attached. Okay, so anything that has this is considered a steroid. Now you might be looking at this and be like, yeah, that looks really nonpolar. It's a hydrocarbon, it's a bunch of carbons and hydrogens, not very soluble in water. And that's why steroids kind of fall under lipids. Okay, so a type of steroid, one is cholesterol, which I am sure you have all heard of. It is the most important and the most abundant steroid in the body, and it's the basis for many hormones. So this is a cholesterol molecule. Notice the 6 carbon rings and the 5 carbon ring there. And then it's got this group, this acyl group on here. It's got a double bond down here. It's got an OH over here. So there's some modifications to a generic steroid, but this is what cholesterol looks like. And then we can make cholesterol into some really important hormones like estrogen and testosterone. Now, there's not huge differences between these molecules. I've kind of circled where the differences are, right? This is an aromatic ring with an OH on it, and this one is not aromatic. It's just got a double bond with a double bonded O and a methyl on here, but the structure is still a basic structure of a steroid. So these kind of fall under that steroid category. Now, lipids are very nonpolar. At least part of the molecule is going to be nonpolar. So how do they move throughout the body if the body is made of mostly water and polar materials? Well, they are transported via what is called a lipoprotein. This is a lipoprotein. Okay, they are transported as lipoproteins. So there's 2 different types. There's low density lipoproteins and high density lipoproteins. And what it is it's a big ball of lipids. So all of my lipids go in the inside of this ball because they don't attract very well to water, right? So there's a core that has our triacylglycerols or our triglycerols or whatever you want to call them, all of those things that have either 3 nonpolar tails, or some of them are a polar head with 2 nonpolar tails. Cholesterol esters, all kinds of stuff that doesn't like water goes on the inside. And then the outside is made by phospholipids and apolipoproteins, which are really just proteins that are more attracted to water. So then this ball can kind of transport through the body to get those lipids where they need to go. So a lipoprotein is a sphere with those proteins on the outside, and it'll take those where they need to go. Okay, you don't have to know the semantics of this picture here. It's just showing you kind of that we've got cholesterol involved, like in your artery walls, right? And then we can transport that cholesterol via an HDL or a high density lipoprotein. And that's where their cholesterol, it gets turned into esters, and we can move it wherever we need it to go. Okay, and these are the differences between high density lipoproteins and low density lipoproteins. So as you notice, the good or high density lipoproteins are small. Okay, that's very dense. You have those particles moving. You don't have a whole lot of unsaturated fatty acids, and those are called the good HDLs. Okay, the bigger they are, the less dense they are. They're bad or not as good for you. If you have a lot of these, you probably have like high cholesterol issues like that. So this is where different cholesterols come in and those lipoproteins going on in your blood.